Friedreich's ataxia is a disorder where there's impaired mitochondrial function that results in damage to various organ systems. In particular, the nervous system gets damaged, which causes ataxia, where the muscles cannot be moved in a coordinated way. The disorder also affects other organs like the heart and pancreas. The disease gets its name from the German physician Nikolaus Friedreich, who first described the disease over 150 years ago. So normally on chromosome 9, there's a gene called the FXN gene that encodes a mitochondrial protein called frataxin. The normal amount of frataxin varies by tissue, with some tissues like the nervous system, pancreas, and heart containing lots of it. Frataxin helps put together cofactors called iron-sulfur clusters. It is a combination of iron and sulfur that form part of enzymes with many functions, such as electron transfer, which is a key part of mitochondrial ATP production. Friedreich's ataxia is caused by a mutation in the FXN gene where there's an abnormal repetition of a GAA sequence within that gene. This is called a triplet repeat, or trinucleotide repeat, which means that a group of 3 DNA nucleotides is repeated multiple times in a row. In this case, guanine, adenine, and adenine. Normally, the GAA sequence is repeated 7 to 34 times within the FXN gene. But in Friedreich's ataxia, there is a repeat expansion where there are 100 to 1,700 times as many copies, with most individuals having repeats ranging from 600 to 1,200 times. Now, Friedreich's ataxia is inherited as an autosomal recessive condition. It's passed on by parents who are carriers because they have one expanded FXN gene and one normal FXN gene, but they don't have any symptoms of Friedreich's ataxia. They end up passing on their expanded FXN genes to their kid. Inheriting both copies of the FXN gene with an expanded GAA repeat is the most common way to get Friedreich's ataxia. The repeat expansion causes gene silencing, which is when the FXN gene is not transcribed normally, and very little frataxin protein is made. With low levels of frataxin, the mitochondria are unable to efficiently incorporate iron into iron-sulfur clusters, and as a result, there is lower mitochondrial ATP production. So there's less energy available for the cell. Furthermore, iron accumulates inside the mitochondria, which reacts with oxygen to create unstable oxygen radicals. Over time, these free radicals damage DNA and proteins in the cells in a process called oxidative damage. Then, this energy deficiency and oxidative damage result in dysfunction and death of the cells that are highly dependent on mitochondrial function, such as neurons, cardiomyocytes, and pancreatic beta cells. The loss of neurons leads to ataxia. The ataxia usually begins in childhood, which causes them to have difficulty in walking. They might have some muscle weakness and loss of proprioception, which is the sense of your position in space relative to other objects. Usually, it affects the legs and then moves up to the torso. A generalized feeling of fatigue is also common. Eventually, most people with Friedreich's ataxia need a wheelchair to get around. The diagnosis of Friedreich's ataxia is established by genetic testing. This test is used to identify the expanded GAA repeat in both copies of the FXN gene, the most common cause of Friedreich's ataxia. Additional tests may be performed to help determine the extent of the disease, such as an MRI of the brain and spinal cord.